Welcome to section 3.4. Now we're going to look at unique characteristics of eukaryotic cells. In this section, we'll look at what makes eukaryotic cells distinct, look at their internal versus external structures, identify organelles that are unique to them, compare them to prokaryotic cells, and we'll finish with a review of how eukaryotes divide compared to binary fission in prokaryotes. Eukaryotic cells are generally larger than prokaryotes, so typically between 5 to 20 micrometers larger. Their most important and largest feature is usually the nucleus, which is surrounded by a nuclear membrane or a nuclear envelope, and this houses the linear chromosomes, and these chromosomes are wrapped around proteins called histone proteins. Unlike prokaryotes, eukaryotes also have membrane-bound organelles like mitochondria and chloroplasts, and these cells divide by mitosis or meiosis, not by binary fission like we saw in prokaryotes. Eukaryotic ribosomes are 80S, they're larger, and they're found in the cytoplasm and studded on the rough endoplasmic reticulum. But they also have 70S ribosomes in those mitochondria and chloroplasts. There are free ribosomes, so the free ribosomes that are floating in the cytoplasm, they are responsible for producing proteins that will stay in the cytoplasm, so water-soluble proteins. Whereas the membrane-bound ribosomes, the ones that are studded on the rough ER, are going to be either exported out of the cell or eventually stuck in some kind of membrane. Here is a general picture of eukaryotic cell. You can see this large nucleus. It's much larger and more complex than prokaryotic cells are, and we see a lot more membrane-bound structures than we do in prokaryotes, since they did not have any membrane-bound organelles. Unlike prokaryotes, which have primarily spherical or rod-shaped cells, for eukaryotes, their shapes really vary. And some shapes include spheroid, cuboidal, fusiform, which means spindle-shaped, and there are many, many others. The shape is influenced by the cytoskeleton, but also by the environment. We just saw that giant nucleus in the cell picture. This is where DNA is stored. Transcription also occurs here, which is when you read DNA and produce messenger RNA, and heredity controls, since our chromosomes are located here. Within the nucleus, you also have a usually dark structure called the nucleolus, and there can be more than one inside a nucleus. In that case, it's called nucleoli. This is the location of ribosomal RNA, and also the assembly site for ribosomes. In the picture on the left here, I have the nucleus, and I can see the nuclear envelope. I'm going to try to trace it, kind of. Oops, a little bit off, but close. That's the nuclear envelope, the boundary, and I can tell because here we have ER, the endoplasmic reticulum. I can see those wavy lines. Here we have our nucleolus, where ribosome synthesis begins. On the right, they've highlighted the nuclear envelope in bright green, and the nuclear lamina is a set of intermediate filaments that maintains the shape of the nucleus. So it's right underneath, right inside of the nuclear envelope. Here are very similar pictures. I can see my nucleolus here. I have my nuclear envelope, and then you can see the rough ER out here. Same thing, rough ER outside here, those wavy lines, my nuclear envelope boundary, nucleolus. Right outside of the nucleus, we have our endomembrane system, and the endomembrane system consists of the ER, the Golgi, lysosomes, and vesicles. For the ER, or endoplasmic reticulum, we have two subtypes, although they're made of the same membrane. The rough ER is the one that is studded with ribosomes, and their job is to produce proteins for export, or that will be embedded in some kind of membrane. The smooth ER, in contrast, is responsible for producing lipids and also for detoxification. Interesting example could be looking at a normal person compared to a person who might have had alcohol addiction. If you look at their liver and look inside their liver cells, the person who consumed a ton of alcohol will have much more smooth ER in their liver cells than the normal person. The next one, the Golgi apparatus, is responsible for modifying proteins and lipids. It's also often known as the post office of the cell because it's responsible for sorting and packaging and sending these proteins and lipids off to their final destination. Lysosomes contain digestive enzymes. So lysosomes will have different types of enzymes within them to break down macromolecules, and also they can break down pathogens. They can also recycle cell parts or dead cells. For the last one, vesicles, this is a really generic term for a membrane-bound structure that allows things to be shuttled between different compartments. So this can be used for transport. This can also be involved in exocytosis when you're releasing something out of the cell, or secretion of certain molecules outside of the cell. Here's another look at that endomembrane system, which is composed of the endoplasmic reticulum. We have the Golgi here, a vesicle, and a lysosome. These diagrams show the connections between the ER, Golgi, and transport vesicles. Notice how they're right next to each other. So ribosomes that are making proteins for export from the rough ER can simply shuttle their vesicles full of proteins to the Golgi, which modifies, sorts, and packages them and sends them through a vesicle for export. You can see something similar here, although it only really shows you the rough ER portion. For the Golgi apparatus, it looks like a series of stacked discs that are kind of a cup shape. And if I look over here, compared to this picture, you can see there are two faces of the Golgi apparatus. We have the cis face, which is facing the rough ER, and we have the trans face, which is away from the rough ER. Eukaryotic cells also contain peroxisomes, and these break down fatty acids, and they can also neutralize toxins using enzymes such as catalase. So catalase can actually break down hydrogen peroxide so it doesn't damage cell membranes. Mitochondria, you probably hear this a lot, are the powerhouse of the cell. Mitochondria make ATP through aerobic respiration. They have these inner membrane folds called cristae to increase the surface area so they can have a lot of enzymes bound within those membranes. They have their own DNA and 70S ribosomes. Remember, these are similar to bacteria. Chloroplasts are found in plants and algae, and their function is to carry out photosynthesis. They do so through pigments that are embedded within these thylakoid membranes that are stacked into stacks known as grana. So the stacks are called grana. This is plural. Singular would be granum. Here is a peroxisome. It looks like a generic vesicle over here on the right. We can see another one here on the left, the electron micrograph. And in peroxisomes, we have catalase, which is involved in breaking down certain toxins. Peroxisomes can also be involved in lipid biosynthesis. Here's an electron micrograph of our mitochondria. We also have a picture on the left. And the one on the left, you can see the cristae pretty clearly. These are the inner membrane foldings. And the purpose of this is to increase the surface area so you can have more enzymes bound within that membrane in order to produce more ATP. Here is a chloroplast. We would see this in plants or algae. They also have a double membrane, outer and inner membrane, just like mitochondria. Remember, they have also bacterial DNA and bacterial ribosomes, just like mitochondria as well. And I can see those thylakoid membranes where the photosynthetic pigments are bound. They're stacked into a granum here. Granum is one stack. Multiple stacks would be known as grana. In eukaryotes, the cytoskeleton is much more complex compared to prokaryotes and is made of three main fiber types shown here. Microfilaments are the smallest, and they're made of actin. These are involved in moving the cell, providing shape to the cell, and they also enable cytoplasmic streaming, which is when you can see the cytoplasm moving around. So organelles are moving around within the cell. Intermediate filaments are the second largest, or second smallest, you could say. They're right in the middle. They provide stability to the cell. They form the nuclear lamina to maintain the shape of the nucleus. And they can even connect cells through desmosomes, so they can connect cells to each other. The largest are microtubules, which are composed of tubulin. These can form spindle fibers during cell division, like mitosis and meiosis. They can serve as tracks, almost like train tracks, to move organelles around the cell. So let's say this is a long microtubule, and I have an organelle here. I can use the microtubule to move it from the left side of the cell all the way to the right side of the cell. Microtubules also make up cilia and flagella. For the spindle fibers, I forgot to mention, spindle fibers start from or originate from structures in the cell known as centrosomes. So here we have the cytoskeletal elements, starting with the smallest, microfilaments, which were made of actin. Actin breaks down, or microfilaments break down and rebuild very quickly. So this is important in changing the shape of the cell rapidly. On the left, we have microtubules made of tubulin. These, remember, are involved in the formation of cilia and flagella for movement. They can also be involved in moving organelles from one side of the cell to the next side. On the far right, we have our intermediate filaments, which can be made of different types of proteins. Here we have keratin. These are really for stability. They're for stability in the cell, and they don't break down or rebuild rapidly. They're really kind of maintaining the shape of the cell or shape of structures within the cell, like the nucleus. Here's yet another look at those actin subunits within microfilaments, the smallest type of cytoskeletal element. These are involved in amoeboid movement, kind of like a slimy movement, cytoplasmic streaming, where the cytoplasm is moving around, even involved in contractile ring formation. During cell division, mitosis and meiosis, you see the cells split when actin forms a cleavage furrow between the cells. And in anatomy, you learned about actin and myosin and how they work together for muscle contraction. Intermediate filaments are known for their stability, so they're involved in maintaining the shape of the cell, holding things in place. For example, in that nuclear lamina, we have intermediate filaments, or in structures that hold cells together, like those desmosomes. These are very stable, preventing two cells from tearing apart from one another. Here we have microtubules, the largest of our cytoskeletal elements, made of tubulin and involved largely in movement. And movement can be through cilia or flagella, or microtubules can act as train tracks like we see here, and it's moving something like a vesicle or some kind of organelle from one side of the cell to the next side of the cell. Centrosomes that you see in eukaryotic cells and are involved in the organization or arrangement of chromosomes during mitosis and meiosis are also composed of microtubules. So if I look closely at the centrosome, you can see they're composed of triplets, threes. See these microtubules arranged in threes, and there are 9 triplets that make up each of these centrioles. The plasma membrane of eukaryotes also follows that fluid mosaic model, but some differences include that the membrane of eukaryotes can contain sterols like cholesterol, and this is for stability for the cell membrane. Eukaryotes can also easily bring in material through endocytosis, and this can be through phagocytosis, which is when you're bringing in large molecules, cell eating. Pinocytosis is when you're bringing in small solutes or liquids, cell drinking. Or receptor-mediated endocytosis. This is when a substance has to bind to a protein on the surface of the membrane, and then the membrane will invaginate and it'll take the substance in. Molecules or things can be released through exocytosis. And in eukaryotic cells, their cell walls are not made of peptidoglycan. They can be made of cellulose, like in plants, chitin for fungi, silica, and others. We'll see a bunch of examples in Chapter 5. There are no cell walls in animal cells, but we do have an extracellular matrix, or ECM. Sometimes we abbreviate it that way. And that is made of proteins and carbohydrates that provide support and can transmit signals into and out of the cell. The eukaryotic plasma membrane is similar to prokaryotic membranes, but here we also see the addition of sterols like cholesterol for stability, and usually we'll have a greater variety or more glycoproteins and glycolipids that are important in cell identification and recognition of other cells. Here are different types of endocytosis. We have cell-eating phagocytosis, where large particles are being taken in, and you can see that vesicle or vacuole formation. Here we have cell drinking, where small solutes are being taken into the cell. And then receptor-mediated endocytosis is when the substance has to bind to a membrane-bound receptor before the lipid membrane will invaginate, forming this coated vesicle. Here we have the extracellular matrix that can protect the cell from physical stress. It can also transmit signals through changing the shape into the cytoplasm, so into the cell. We saw earlier that some prokaryotes have flagella. Eukaryotes can also have flagella. In this case, they don't rotate like a propeller. Instead, they're more of a whip-like movement. They're always arranged in a 9+2 microtubule arrangement. So microtubules are what make up eukaryotic flagella. Microtubules also make up cilia. Cilia are usually shorter. There are usually many more on the surface of the cell. They can also be involved in moving the cell, but also sweeping things across the surface of the cell. Both of these can be powered by motor proteins known as dynein. Here's another look at eukaryotic flagella. If I take a cross-section of the filament of the eukaryotic flagellum, you can see that there is a 9+2 arrangement. That means there are 9 pairs of microtubules on the outside edge, and then 2 single microtubules in the center. So this is true for the most of the flagellum, the filament part, but where it's attached to the cell membrane, the basal body, there is a triplet arrangement similar to what we saw in the centrioles of the centrosome earlier. Another difference between eukaryotes and prokaryotes: while prokaryotes divide through binary fission, producing identical cells, in eukaryotes there are 2 types of division. Eukaryotic cells can divide by mitosis or meiosis. Mitosis will produce 2 identical diploid cells, and it's used when cells need to grow or repair themselves. Meiosis, in contrast, produces 4 unique haploid cells or gametes, and it's key for sexual reproduction. Both of these involve DNA replication, chromosome segregation, and spindle fibers, unlike prokaryotic binary fission, which is very simple and much quicker. Binary fission in prokaryotes is summarized here. First, the prokaryote makes a copy of its DNA or chromosome. Then the cell elongates, and the chromosomes begin to move to the opposite ends of the cell. The cell membrane and wall invaginates, forming a septum between the 2 chromosomes. Eventually, the septum will be complete, and the cell pinches in 2, forming 2 daughter cells. If the cell stays connected to each other, this leads to a diplo or paired arrangement, and with repeated binary fission, this could form a chain or strepto arrangement, but they can also separate, forming a single arrangement or single cells. Here we have another checkpoint. Let's look at differences between eukaryotic and prokaryotic cells. Eukaryotes have a nucleus with linear chromosomes, whereas prokaryotes have a nucleoid with circular DNA. Eukaryotes also have membrane-bound organelles like the ER, the Golgi, the mitochondria, whereas the prokaryotes do not. Eukaryotes have 80 S ribosomes, whereas prokaryotes have 70 S ribosomes. Components of the endomembrane system: these include the endoplasmic reticulum, the rough ER, which makes proteins, the smooth ER, which produces lipids and detoxifies. We have the Golgi, which is the post office of the cell, which modifies, sorts, and packages proteins. Lysosomes digest macromolecules and pathogens. We have vesicles that transport materials between compartments or in and out of the cell. Looking at the different types of cytoskeletal structures, we talked about microfilaments produced or made of actin. These are the smallest, and they provide shape, motility, and cytoplasmic streaming inside of the cell. Intermediate filaments, which were medium-sized and provided structural stability. They also form the nuclear lamina and anchor organelles. We have microtubules made of tubulin. These are the largest. These are hollow tubes. They can form the mitotic spindle, move organelles around. They also make up the cilia and flagella. All three of these work together to support cell shape, transport, and cell division, but they differ in their size and specific roles. If we look at the cellular envelope of eukaryotic cells, that membrane, how does that compare to prokaryotes? In eukaryotic plasma membranes, it's also a fluid mosaic, but there are sterols like cholesterol that add structure and rigidity to that plasma membrane. In eukaryotes, we're also capable of endocytosis and exocytosis. For eukaryotic cell walls, there are walls in things like plant cells and fungi, but animals do not have cell walls. Algae can have cell walls, and their makeup really varies. For prokaryotes, they have a plasma membrane and a peptidoglycan cell wall, which can be thick or thin. Archaea also have a cell wall, but it's not made of peptidoglycan. Instead, they have pseudopeptidoglycan. And other differences, prokaryotes use peptidoglycan, whereas eukaryotes use polysaccharides like cellulose or chitin in their walls. For the last one, differences between flagella in eukaryotes and prokaryotes. Eukaryotic flagella are flexible and they have a whip-like motion, and that have that 9+2 microtubule arrangement, whereas prokaryotes, their flagella are rigid and they rotate like propellers, and they're anchored by the basal body. Both of them provide motility, but they differ in their structure, their protein composition, and how they move. I cut and paste the chart from our book into our slides. We have prokaryotes here. Prokaryotes include the bacteria and archaea versus the eukaryotes here. Take a look and be able to compare and contrast them. We can see, for instance, eukaryotes are much larger. They have multiple chromosomes. They have histone proteins. And archaea sometimes can also, because they're more similar to eukaryotes than prokaryotes are. Look at division. They divide through binary fission for prokaryotes, whereas in eukaryotes we have mitosis, meiosis. We have sterols in the cell membrane. The table continues on this slide. Bacteria, remember, have peptidoglycan in their cell walls. There are a few that don't have cell walls. For flagella, we talked about flagella and cilia being composed of microtubules and eukaryotes. And remember, there are no cilia in prokaryotes, only in eukaryotes. Only eukaryotes have membrane-bound organelles and thus the endomembrane system. Ribosomes are different in prokaryotes and eukaryotes, and that is why we can use antibiotics to target these prokaryotic ribosomes, although there might be side effects because they are also present in our organelles like mitochondria and chloroplasts. All right, let's recap 3.4. Eukaryotic cells are larger and more complex than prokaryotes. We have a nucleus with nuclear membrane and linear chromosomes, membrane-bound organelles like the ER, Golgi, mitochondria, chloroplasts, lysosomes. Eukaryotes also have 80 S ribosomes in the cytoplasm and on the rough ER, but also 70 S ribosomes in mitochondria and chloroplasts. We also have a cytoskeleton, either microfilaments, intermediate filaments, or microtubules for shape and transport. Eukaryotes reproduce by mitosis or meiosis, but not binary fission. Looking at the internal and external structures, we looked at internal structures like the nucleus, which stores DNA, the nucleolus, which is involved in ribosomal RNA synthesis. We have the ER, the rough ER is involved in protein synthesis. The smooth ER is lipid synthesis and detoxification. The Golgi was for modification, packaging, and secretion of proteins. For lysosomes, we had digestion. Peroxisomes detoxify. Mitochondria are involved in ATP production through aerobic respiration. Chloroplasts are involved in photosynthesis in plants and algae. And the cytoskeleton was for shape, motility, and transport within the cell. For the external structures, we have the plasma membrane, we have the cell wall in plants, fungi, and algae. Remember, animal cells do not have cell walls. We have the extracellular matrix in animals that is composed of proteins and carbohydrates for adhesion and signaling. And we have appendages like flagella and cilia. All right, looking at structures, organelles, unique. So we had the nucleus, the endomembrane system. Remember that was made of the ER, Golgi, vesicles, and lysosomes. We had mitochondria and chloroplasts. Cilia, right? We don't have cilia in prokaryotes for moving and sweeping functions. We have that cytoskeleton, the extracellular matrix. Looking at similar structures between prokaryotes and eukaryotes. So genetic material, they both have DNA. Prokaryotes have a circular chromosome in the nucleoid. Eukaryotes, though, have a linear chromosome in the nucleus. Both have ribosomes, but differ in size. Right? 70S in prokaryotes, 80S in the cytoplasm for eukaryotes. Both can have cell walls, but in prokaryotes, they're made of peptidoglycan and bacteria, whereas eukaryotes use things like cellulose, chitin. They both have plasma membranes, but eukaryotes have sterols like cholesterol in their cell membrane, and they can perform endocytosis and exocytosis. Both have flagella, but in prokaryotes, the flagella are rigid and rotate like propellers, whereas in eukaryotes, they're flexible and whip-like, and they have that 9+2 microtubule arrangement. For organelles, prokaryotes, we're going to learn more of this later, but they actually do metabolism in the cell membrane, whereas eukaryotes perform metabolism mainly in the mitochondria and chloroplasts. And then looking at this last one, remember that eukaryotes can undergo mitosis and meiosis. In mitosis, they're producing two identical diploid cells for growth and repair. In meiosis, you're forming 4 genetically unique haploid cells, and that's usually for sexual reproduction and gamete formation. Whereas in prokaryotes, we have binary fission. That's very simple. You copy your DNA, the cell elongates and divides into 2 identical cells. Binary fission is easy because there's no nucleus involved, no spindle fibers, and it's much faster than mitosis and meiosis. And that takes us to the end of Chapter 3. I'll see you next time in Chapter 4.